a lot for this very kind introduction. I will try to find my slides, which are here. Okay, so thank you very much for inviting me, in particular, Jack. I'm very happy to see you again. And so I'm going to talk about emerging antibiotic resistance in 2023, and mostly in gram negatives. Why? Because the two other main problems worldwide are very well identified, and in many areas in the world, including in Switzerland, very much controlled. I'm thinking about MRSA and VRE. So I'm just going to talk just a little bit about Enterobacteriaceae, Pseudomonas, and Acinetobacter baumannii. And as an introduction, I shall underline that the problem today and for the next 30 years, I will not be there. Within the next 30 years is going to be on Enterobacteriaceae. Why? Because E. coli still remain the main culprit in terms of infection for humans and antibiotic resistance. So for the young generation, focus on E. coli. And one of the best examples is the spread of ESBL producer in E. coli since I would say 2000. And you can see here a very traditional antibiogram by this diffusion, and you can see multi-drug resistance, including resistance to extended spectrum cephalosporin and non-beta-lactams antibiotics. So this story is very well known, and in a way, for non-determined reasons, quite controlled. And you can see here the slide about what's going on in Switzerland. You can see that in invasive E. coli and Klebsiella pneumoniae in Switzerland, since I would say 2015, we are reaching a plateau and some kind of decreasing number of ESBL producers in E. coli. Why? We don't know exactly. Because in Switzerland, like in all of the parts of Europe in the world, we didn't change so much the use of antibiotics. Why? This is a way of the nature. And you can see that it's exactly the same trend in E. coli in Europe. You can see more or less a plateau, and in some countries like Italy, we have even a decrease, which is quite surprising. But there are facts that have been controlled by the so-called Center for Infectious Disease, European Center for Infectious Disease in Stockholm, that gave us all the data on that. And if we have a look in the world, this is a broad, spectrum of the spread of E. coli, ESBL produced in the world, we can underline that the epicenter of the problem still remain Asia, and mostly Pakistan, India, and Bangladesh, and this part of the world. And the reasons are very simple. Overuse of antibiotics, no control or very few control of hygiene in many areas of the health system in this part of the world. But by the way, the most important problem are the spread of the so-called carbapenemesis that confer resistance to all beta-lactam, to make a long story short, including carbapenem. So the very first have been reported from the US, from the east part of the US, and the author called this carbapenemas KPC because it's mostly reported in Klebsiella pneumoniae. And you can see some kind of resistance to all antibiotics except fosfomycin, colistin, and some strains, gentamicin. The current situation of this KPC producer is the following. Still quite important in the US, quite important in South America, in India, and in Europe. What we are seeing, this KPC producer, mostly in Klebsiella pneumoniae and mostly in the east part of Europe. And I am thinking about Poland, Hungary, and Bulgaria. In the other parts of the world, and I would think in Switzerland, the situation for KPC producers is quite controlled. Why? Because these KPC producers are mostly identified in Klebsiella pneumoniae. And Klebsiella pneumoniae is mostly a hospital-acquired pathogen. It's not a community-acquired pathogen. So when we have an outbreak with that, this is, in a way, quite simple. Meaning, we identify the problem, we screen the patient, we isolate the patient, and then we can control an outbreak. So the KPC producer in 2023, still in the world, are mostly in Klebsiella pneumoniae. And this is a question of the willingness of the people, the doctor, and all, to control the outbreak, because it is mostly located in the hospital. This is not the case of the second most important, carbapenemase, that is NDM, standing for New Delhi metallo-beta-lactamate. Because the first report, and this is the first paper on that, has been done by people located in Sweden, together with British people with strains coming from India. And you can see here again some kind of resistance to everything except colistin and fosfomycin. The current situation of this NDM, they are spreading worldwide at very high level. And what we are observing in Europe and in other parts of the world, the spread of E. coli producing a very peculiar NDM, which is NDM5, which has more activity in terms of carbapenem hydrolysis than the other NDM, such as NDM1 and NDM2. And this is a source of concern because E. coli is mostly a community-acquired pathogen in Switzerland, like in other parts of the world. And as mentioned before, everything which is located in the community is just impossible to be controlled. People are talking about antibiotic treatment. Community, we cannot do anything. We just see, identify, but the spread, in particular for E. coli, very difficult to control because the very first things we are doing every morning is to shake hands to friends and family. And then by doing that, we exchange at least E. coli. So very difficult to be controlled. So I am a little bit concerned about the following fact. The spread of the plasmid carrying NDM5 from E. coli to Klebsiella pneumoniae. We have seen that very much for what I was just saying before, ESBL. This is a source of concern for the next couple of years. And again, where is the epicenter of this NDM producer? India, Pakistan, Bangladesh. I have to point that, according to the latest data in India, they estimate that the rate of resistance to carbapenem in E. coli in India is around 18%. For giving you a comparison, in Switzerland, this rate of carbapenem resistant E. coli is 0.1%. So huge difference. And so this is clearly a source of concern because those trends are spreading all over the world. The third and latest, carbapenemase I'm going to detail today is the so-called OXA48. And OXA48 we identify in Paris with a strain coming from Turkey. And after that, it has been identified in many areas in the world, but mostly in the northern part of Africa and Middle East and Turkey. And this OXA48, which is a peculiar carbapenemase, its gene is almost always located on a single type of plasmid, which is so-called 62 kb plasmid, and that spread at a 10 to 100 higher fold rate than a regular plasmid from Enterobacteriaceae. Meaning, this is the reason why in a given patient, we can observe in the flora, in the gut flora, OXA48 in E. coli, on Tuesday, Klebsiella pneumoniae, on Wednesday, cloacae, and on Saturday, Proteus mirabilis. This is not mistake from the lab. This is really exchange in the gut, because, in the gut, this is a very nice, flora to spread, the antibiotic resistant genes. Because here you have something to eat for the bacteria. It's warm and it's quite stable mostly during the day. Perfect for exchange. When we are doing mating out, meaning conjugation experiment in the lab, that's exactly what we are doing. We mix two bacteria, we wait a little bit of time, we put that at 37 degrees and we wait 18 hours. And if we are lucky, we have transfer from a plasmid from one bacteria to another. Exactly the same for the gut. And so this explains the quite wonderful success of this OXA48 gene within the last 10 years. And where this gene is located, mostly in the Middle East. And when I was in my previous life, when I was working in Paris, I asked my secretary, she was in vacation and she wanted to go on vacation in so-called Club Med, in Morocco. And I told her, please give us some ground sample from the ground, and we will look for OXA48. And we identified plenty of serratia with this OXA48, meaning already, in 2011, these genes was spread very much, in the ground. And for the funny story, when we submit that to antimicrobial agents chemotherapy, the editor asked me, which is the statistical design of that? There was no statistical design. I just ask my secretary, please give me some sample. So I justify, I was in between, but it has been published. And so in Switzerland, we identify the spread of plenty E. coli, strains, but to make a long story short, most, if not all the strains carry the same plasmid carrying the same OXA48. So it is very difficult to trace an outbreak of OXA48 due to this fact. And another thing which is quite interesting about the OXA48-like strains, sometimes such variant of OXA48, such as OXA244, they produce a very specific carbapenemase that confer very low level of resistance, if any, to carbapenem. Meaning, you can see on that slide, here, within the square, the yellow square, you have meropenem, imipenem, and ertapenem. The three are working quite well for against OXA244. Meaning, in a regular clinical lab, it is quite impossible to detect those OXA244. Meaning, they spread silently within the community. And the current situation, about in Switzerland is the fact that we are seeing an increasing of this number of strains that produce carbapenemase. Another interesting fact related to OXA48, but we have observed with other type of carbapenemase producer, is the fact that some Klebsiella pneumoniae are so-called hypervirulent. The definition of hypervirulent strains is a clinical definition, which is sustained by the presence of a series of virulence genes. What the following situation, we have observing the following situation. In some cases, we identify virulence genes within strains that are not obviously virulent. So the correspondence in between identification of the so-called virulence genes and the clinical state is difficult to match. But for sure, and this is exemplified by these strains we identify with the people from Lausanne, this strain came from a Russian patient who have liver disease related to this Klebsiella pneumoniae strain. Let's switch rapidly to the story of Pseudomonas aeruginosa. Pseudomonas aeruginosa mostly is like Enterobacteriaceae. It possesses the same type of resistance pattern and multi-drug resistance pattern, to make a long story short. One of the latest reports is the problem of colistin resistance in Pseudomonas aeruginosa. You know, many people think that colistin, which is a very old antibiotic dating from 1963, could be interesting for treating patients when we are in front of multi-drug resistance bacteria. It is very much debatable because it is quite nephrotoxic, and second, it does not diffuse very well in tissue. So whatever, it is used in many parts of the world, including in the US. And it has been used in the US to a large extent. And if we look to the resistant Pseudomonas to polymyxin, this is a map, and you can see, you know, the situation in the US, which is incredibly high for Pseudomonas aeruginosa. In the other part of the world, either we have no data, but I can tell you that in Switzerland, according to the strain we are receiving in this reference center, the strains are mostly susceptible to polymyxin. An interesting other, you know, problem in terms of emerging antibiotic resistance is the so-called methylases. You know, these methylases modify the RNA. Therefore, the aminoglycoside cannot bind anymore to RNA. Those methylases confer pan-drug resistance to all aminoglycosides, as opposed to the old, you know, type of antibiotic resistant genes that modify the aminoglycosides themselves. And so this has been reported by the team of Patrice in 20 years ago now. And the very first report was from Lucerne 5 years ago. And you can see here, you know, this resistance to amikacin, netilmicin, and kanamycin. And this double zone is something very much in favor of presence of a methylase. And this has been reported by a series of groups, including the group of Jacques Schrenzel here, or Andrea and Demiani in Bern. And those genes are spreading very highly, meaning they may kill the efficacy of aminoglycosides in Enterobacteriaceae, Pseudomonas, and Acinetobacter baumannii. And this is a huge source of concern. And in all meetings, people are talking very much about beta-lactam. But we have novel beta-lactam. We don't have any novel aminoglycoside. And you know, these antibiotics are very much efficient for treating any infection rapidly, including urinary tract infection. And so what we have observed, you know, within the last 4 years here in Switzerland, a quite increasing trend of these methylases in Enterobacteriaceae. What about multi-drug resistant Acinetobacter baumannii? First, the WHO mentions that Acinetobacter baumannii is a very important multi-drug resistance bacteria, which is true. But in terms of number of infections, this is not true at all. Still today, Enterobacteriaceae are by far the most important number of infections in hospital. Acinetobacter baumannii is a problem because it persists in the environment much more than E. coli and Klebsiella pneumoniae. And we don't know why. But this is a fact. When we have an outbreak of multi-drug resistant Acinetobacter baumannii, we are in problem. And you can see here one example of this multi-drug resistance producing the so-called OXA23, which is a peculiar carbapenemase specific to Acinetobacter baumannii. And the worldwide situation in multi-drug resistance is quite important in Acinetobacter baumannii. You can see that in South America, Central America, all the Asia, you know, including northern part of Asia, meaning Russia, from Russia to India, there are very huge, you know, high rates of resistance to carbapenem in Acinetobacter baumannii. And same in Morocco, Libya, Algeria, same in North Africa, same in Middle East. So here, for the future, in terms of importation of strain, for example, for Switzerland, this is a source of concern. And what we are seeing, in our reference center, huge number of this multi-drug resistance Acinetobacter baumannii. And Acinetobacter baumannii has acquired NDM gene, and this is an example of one of these bacteria. And from a genetic point of view, this slide for me is quite interesting. Is the fact that we still don't know the natural reservoir of NDM. What we have evidence is from different genetics arguments. This gene has been spread to Acinetobacter baumannii, which is an intermediate host to spread this gene to Enterobacteriaceae. And this is, I would say, a quite different concept of what we think. Meaning, we think that most of the important antibiotic resistant genes are located, in Enterobacteriaceae and then spread to Pseudomonas on Acinetobacter. For the NDM, it is very likely the opposite. In terms of resistance to novel beta-lactam, for sure we have novel antibiotics, which is an excellent news. People are saying, in all the news, we don't have any novel antibiotic. This is just wrong. We have wonderful novel antibiotics, such as ceftazidime, aztronam, meropenem, vaborbactam, imipenem, rolobactam, cefiderocol. Just to mention those that are on the market, who are going to be very soon on the market, such as aztronam, avibactam. And they may target, many, if not all, carbapenemas produced. So this is an excellent news. The problem, we are seeing a few antibiotic resistance bugs that are resistant to these novel antibiotics. And one of these examples is this one, the so-called KPC3, which is a novel carbapenemase. And in some strains, it has been observed the selection of derivative of KPC2 or KPC3, such as KPC41, that confer resistance to Cephta avibactam. But the excellent news for this most, if not all, of this KPC derivative, they confer some kind of susceptibility to carbapenem. Meaning that we can think using carbapenem for treating this KPC41 producer. And second, from a clinical point of view, people are still saying, oh, novel antibiotic, let's keep this novel antibiotic for rainy days when I don't have anything else. Meaning, when we have a KPC producer in Fribourg, like in Geneva or in Lucerne, what we are doing? We are using ceftazidime plus avibactam. Maybe this is an important mistake. Maybe we shall use meropenem, vaborbactam, or imipenem, rolobactam, because it's much more difficult to obtain those type of mutants than by using ceftazidime avibactam. And in addition, many of these ceftazidime avibactam resistant mutants confer resistance to one of the latest antibiotics, which is cefiderocol. And here is one paper which is in press, indicated the huge number of variants of KPC that confer resistance to ceftazidime avibactam. And so you can see this number is increasing at a very high rate. And here in Europe, the epicenter of the problem is clearly Italy. Italy, they have used a lot of the ceftazidime avibactam, and they have selected a lot of this KPC producer. And those type of patients, I would say, are not very much in control in this country. In Switzerland, the story of KPC producer is very much controlled because there is screening of the patient around 1 case. In terms of resistance to cefiderocol, complicated issue. But to summarize, we have either modification of the transport of the cefiderocol inside the gram-negative bacteria, cases in Acinetobacter pseudomonas. We have modification, we have acquisition of a series of beta-lactams, such as NDM and PER, that confer higher degree of resistance to cefiderocol. And one of the latest examples of novel antibiotic resistance is resistance to Astrionam avibactam, which is under development in Pfizer in the US. And this resistance is mostly observed in E. coli. It is associated to modification of the target of Astrionam, which is the so-called penicillin binding number 3, penicillin binding protein number 3, plus a carbapenemase of NDM type, plus a cephalosporinase CMY2 or CMY42. And together with our friend from Paris, my colleague and friend, Professor Ruppe, we have identified that those Astrionam avibactam resistant strains have been identified in many countries in the world, including in France, in Switzerland, and in Pakistan. And we identify very identical clone in Switzerland, in France, and in Pakistan, meaning that the source is clearly Pakistan in that case. And so to end my talk and to be just on time, because we are in Switzerland, I should underline the following fact. For the COVID, the control of the COVID was quite easy. We have an outbreak, people were sick, they were isolated, and everything went in control. So the story of acute outbreak is easy in a way. Here, what we are facing is an outbreak which is very difficult to identify with a different clone, different antibiotic resistance. And so in that case, this is a so-called outbreak, silent outbreak, which is very identical to control. And this is the reason why the problem for the next of antibiotic resistance for the next 20 years will be an important issue worldwide because of the diversity of the clone, diversity of the antibiotic resistance genes. So I thank you very much for your attention.